welcome to this hands-on demo of PanHunter. In this video, we will show you our capabilities in the field of single nucleus and spatial transcriptomics. Everything you will see is happening in real time. Chronic kidney disease, short CKD, is a global health threat, as it affects 1 out of 13 individuals. There are more than 600 million CKD patients worldwide. Accordingly, scientists here at Evotec are looking into better ways to understand and treat CKDs. To do that, they build and use a common model in renal research, ischemia-reperfusion injury, short IRI, to reproduce the CKD phenotype. IRI is an injury of the tissue that results from blood and oxygen returning to a tissue area that was cut off from both for a critical period of time before. The specimens in this study are untreated and IRI-induced kidneys of 8-week-old mice. The kidneys were dissected 6 days after inducing the ischemia. Subsequently, 2 datasets were generated, one from untreated and one from IRI kidney, using 2 different approaches, spatial transcriptomics and single nucleus transcriptomics. Both datasets were generated within our internal PanOmics study using the Visium spatial and Chromium single-cell gene expression platforms, both from 10x Genomics. In the next couple of minutes, we will look for marker genes of inflammation, kidney injury, and fibrosis and their regulation. Afterwards, we will combine both datasets to gain higher resolution and look for spatial differences on the transcription level across the kidneys. Finally, we will try to find individual stages of disease progression. Okay. So now let us open PanHunter and directly go into the single-cell browser, which is our app to investigate single-cell, single-nucleus, and spatial transcriptomics data. We will start to look at the single-nucleus data from both kidney samples. To do this, we select the respective study and first look at a pre-calculated dimensional reduction plot for all individual cells. In this plot, each dot stands for one cell, and the more similar their transcription profile is, the closer they cluster together. By clicking on the respective entry in the legend, we can switch our selection of cells between untreated and IRI kidney, which will be highlighted accordingly. This way, we can see that there is a strong overlap in general between both samples. The cells form clusters, which usually correspond to common cell types or at least similar molecular profiles. The cell types for these clusters, shown here by color, have already been annotated by our scientists in advance using known cell type enriched marker genes or gene transcription patterns. We can see cells from different regions of the kidney, for example, ascending loop of Henle cells, endothelial cells, or proximal tubular epithelial cells, short PTECs. But for now, let us compare the untreated with the IRI samples and have a look at how they might differ. For this, we switch to a different tab in the single-cell browser, which can be used to compare different samples next to each other. Here, we see a remarkable difference. There are clear changes in size visible for the cluster of the IRI kidneys on the right compared to the untreated ones on the left. These were identified as subcell types with noticeably different activity for important genes using PanHunter and its differential analysis capabilities. The respective cells form a new cluster in the plot, as seen here. To look at this in a bit more detail, we can switch to the Genes tab. Here, let's start with the known marker gene and select the transcription level of COL1A1 to be visualized. This gene encodes for a structural protein, collagen 1, that is involved in scar formation and fibrosis. Hence, upregulation of COL1A1 indicates fibrogenic processes. From this, we can see that this gene is strongly upregulated in one cluster in the IRI study, corresponding to myofibroblasts. Another interesting gene is HAFCO1, known for its relevance during inflammation and immune response. It encodes for the protein kidney injury molecule 1, or KIM1, which is a well-known biomarker for kidney injury. Here, we see that its transcription is upregulated in specific PTECs in the IRI sample, which isn't there for the untreated kidney. This way, we can investigate the differences between untreated and IRI kidneys on the molecular level. Okay, but so much for the different cell types and the transcription levels of individual genes. How can we use this information for a better understanding of the molecular processes taking place in the induced irie kidneys? Here, spatial transcriptomics can be immensely helpful, as it not only allows us to understand the behavior of different cell types or clusters of cells, but it provides the additional information about the localization of those cells within the kidney. Therefore, we now must switch to the spatial transcriptomics dataset for both first the untreated and afterwards the irie kidneys. I will start with the untreated ones. In the background of the plot is an image of an HE stained histological section. In the foreground, we see spots in color representing the transcriptomes of the underlying cells. Thanks to clever cDNA barcoding, we can look at the gene transcription levels for each of these spots individually. The cell type for each spot is defined by the most prominent cell type within this spot. Now let us focus on a single cell type using the annotation from before. Here I have selected all S3 PTECs by clicking on the respective entry in the legend. The localization aligns very well with published data from cell type localization in kidney, which confirms that both our spatial transcriptomics and cell type annotation were carried out successfully. If we use the gene transcription information for individual marker genes and highlight their level of transcription by color intensity, we can see where these genes are most active. When highlighting COL1A1, the fibrosis marker we have already seen before, and compare untreated and irie kidney, we can see an upregulation for irie kidney in the inner core of the kidney. From a biological point of view, this is quite interesting and unexpected, as this is an area mostly occupied by loop of Henle cells, for which one usually would not expect collagen production. For the other injury marker gene, HAFCA1, we see upregulation in the outer ring of the kidney corresponding to the location of PTECs. But let us come back to the unusual upregulation of COL1A1 in the core of the kidney. For this, our bioinformatics scientists use cell type deconvolution, a feature that will be soon implemented directly into PanHunter as well. Cell type deconvolution is a method that uses the transcription information of the isolated cell types from our single nucleus data to reconstruct in silico the mixed expression patterns of individual spatial transcriptomic spots. You could think of it a little bit like trying to remix or reproduce the current color of your wall just from base colors without knowing the exact mixing ratio. By measuring how much of which component color you're using until you've matched the original color, you would be able to reconstruct the recipe for the entire composition. As a result, our scientists were able to get a better picture of the localization of low abundant cell types within the kidney. And indeed, this analysis reveals a possible answer to the question. Myofibroblasts and immune cells invade the core of the irie kidneys, most likely in response to the induced inflammation, and could be responsible for the apparent upregulation of COL1A1 as those cells were clustering together with the loop of Henle cells. We could now go back to the single nucleus data to select individual clusters from the previous plot, or focus on other regions of the kidney, or other genes, or all in combination. Instead, I will go on and run a differential analysis on the clusters with apparent upregulation of COL1A1 to compare them to unaffected cells to confirm our findings also on a larger scale. This analysis, which generates a so-called top table, a list containing all significantly regulated genes, can be started directly from within the single-cell browser. To do this, I select the clusters and conditions I would like to test and start the calculation. Below, I get a preview of the top table, which will be saved to PanHunter by pressing the respective button. To take a more detailed look at the results, we switch to the Top Tables app and look at the gene set enrichment that have been calculated automatically upon storing the list. For this demo, I will look for enriched pathways. Here, we can find the pathways for the electron transport chain and the TCA cycle as being significantly regulated. When we take a closer look, we can see that they are downregulated, which is indicated by the red coloring of the involved genes. This is probably indicating a response to the lack of oxygen and nutrients in the respective tissue. Another significantly regulated pathway is the inflammatory response pathway. When I click it, I see that it's strongly upregulated, shown by the blue coloring, indicating that indeed inflammatory processes are underway. As you can see, PanHunter provides a large spectrum of tools to analyze and investigate your data. Today, I was able to show you only a small part of them. But there is one more thing I would like to show you. For this, let us go back to the single-cell browser and analyze the trajectory of the disease progression, as diseases are rarely binary. For this, we use a combination of specialized algorithms. The first one is FADE, which is used for dimensional reduction. The second one is Slingshot for pseudotime calculation. In the resulting visualization, the cells will separate according to increasing severity of the molecular symptoms. Some of them will show early onset, while others will correspond to later stages of progressing IRI. This way, we could subgroup the different cells into distinct stages of the disease and again investigate their molecular profiles separately. But that's a story for another video. I hope we could give you a very short insight into Evotec's and PanHunter's single nucleus RNA sequencing and spatial transcriptomics capabilities. Thank you very much for watching, and goodbye.